Perfect. So I would just like to start to acknowledging the traditional custodians of the land from which I am joining today. And this is the Ngunnawal and Ngambri people. And I would like to acknowledge and respect their continuing culture and the contribution they make to the life of this city and this region. I would also like to acknowledge and welcome other Aboriginal Torres Strait Islander people who may be attending today's event. So today I'm going to talk about what museums can tell us about biodiversity, evolution, and biogeography, and talk about some new technologies and future perspectives. With billions of specimens in museums worldwide, natural history collections hold an impressive record that is the basis of the documentation of life on Earth. Natural history collections also provide a rich source of material for answering questions in biodiversity, phylogenetics, biogeography, conservation, and many other areas of ecology and evolutionary biology. And CSIRO houses 6 plant and animal collections. We have the Australian National Insect Collection, the Wildlife Collection, the Herbarium, the Algae Culture Collection, the Fish Collection, and the Tree Seed Center. And we send about 200,000 on loan each year to more than 20 countries. And using our specimens, we discover an average of 100 new species each year and engage with hundreds of commercial customers, which 30% of them are international. But our collections are also growing. So we add to them more than 1,200 specimens each year, and we're also making them accessible through digitization and making our records available through platforms like the Atlas of Living Australia. And this is the Australian National Insect Collection is my home, and with 12 million specimens is the largest global collection of Australian insects. But each specimen in our collection holds a vast amount of information. So what's in a museum specimen? Well, basically each specimen in our collection can tell us information from the date and time of collection to where a specimen was collected. And embedded in each specimen, we can have data on the size, the shape, and color. We can establish biotic interactions by looking at the microbiomes, or establish animal-plant interactions looking at the pollen in our specimens. And we also have nucleic acids which can sequence. And now we're looking into sequencing the peptides and looking into the small molecules in our collections as well. And so the last few decades have produced a number of new technologies that are allowing us to uncover more and more information from our museum specimens. And we have high throughput sequencing helping us sequence them much faster and getting larger amounts of data. And we are refining this for degraded DNA sequencing. High performance computing is allowing us to analyze data sets from the data that we get from our collection specimens more efficiently. And at ANIC, we're now developing methods to use mass spec to study the molecules that are present in our collection specimens. And more affordable and portable SEM systems are letting us look much closer into our specimens to study new traits. With metabarcoding, we can establish interactions between organisms. And with artificial intelligence and machine learning, we can obtain traits, identify our specimens, and analyze our sequences much quicker. So what are some applications of these technologies? So Liz Mila and Francisco Encinas Viso are leading this project where we're using the collection specimens and looking at their pollen and doing pollen metabarcoding to establish plant-pollinator interaction networks into the past. And this is along with a large team of scientists from the FSP Environomics project. PhD student Alana McClellan is sequencing parasitoid wasps and their hosts for species delimitation and host coevolution studies. Her presentation is on Wednesday at 3:30 p.m. Postdoc Uday Mawelajedera is extracting the small molecules in our specimens to discover new possible bioactive molecules. And you can watch her presentation, which was yesterday and should be recorded. And postdoc Dan Dashevsky is studying potent wasp venoms to determine their evolution and structure. And his presentation was yesterday. You can also watch the recording. So today I will go deeper into the study of behavioral evolution in spider wasps, the use of museum specimens for a higher level phylogenomics of this wasp family. And I'm also going to talk about some of the behavioral, some of the biogeography of this group. Finally, I will show you some recent results about the evolution of mimicry in velvet ants. So this is the outline of today's talk. And for each topic, I will try to cover these three aspects. So first, let's talk about behavioral evolution in spider wasps. So Pompilidae, family Pompilidae, that are also known as spider wasps, are mostly solitary wasps that use spiders to feed their larvae. There are about 5,000 species described in over 100 genera, and they're currently classified in 5 subfamilies. They are found in all continents except Antarctica, and they have a very, very painful sting. They have been classified as 4 out of 4 in the Schmidt pain scale. And there is a molecular phylogeny, but many of the backbone nodes have low supports. And the oldest fossil we know is 54 million years old. So most of these spider wasps hunt, paralyze, and lay an egg on a spider. And then when the larva hatches, it feeds on the entire spider's body while the spider is paralyzed. But there are many variations to this behavior of hunting, stinging, oviposition, and nesting. For example, some spider wasps look for hosts while the spiders are inside their own burrow. So they go to the spider's burrow and sting the spider, lay an egg on it, and use their own spider's burrow as their nest. So they don't have to make their own nest. They're kind of smart. And then there are others that actually have eliminated the hunting, stinging, and nesting steps by finding another pompilid with an already paralyzed spider and simply going in and laying an egg. So I call these the lazy ones. And these are also known as kleptoparasitoids. So in terms of the host specificity, we have found that pompilids can parasitize spiders from many species and even many families. So they are very generalist at the taxonomic level, but they usually only use hosts of a single or a couple ecological guilds. And we have found that during the course of the evolution of this group, there have been various shifts to the use of different spider guilds. So with these behavioral differences and the difference in the spiders that they parasitize, we wonder if the sensory structures of these wasps are different for kleptoparasitoids and for pompilids that hunt on spiders with different life histories. And this is work that is being done by PhD student from Melbourne Uni, Chris Freelance, and some collaborators. Chris is an expert in scanning electron microscopy, and he used our museum specimens to measure sensory traits for various behavior types within Pompilidae. So we sampled 24 taxa within one of the most diverse Pompilid subfamilies, Pompilinae, and obtained information on their hosts. We observed and measured the antennal sensilla density, head width, compound eye area, and ommatidia using a desktop SEM. And then we used a newly reconstructed phylogeny for this subfamily, which I will talk more about later today, and analyze these variables using ANOVA. And we also compared these structures in parasitoids versus kleptoparasitoids. So first, in terms of kleptoparasites, what we found is that both the compound eyes and the antennae of kleptoparasitoids, the antennal sensilla density is smaller than those of non-kleptoparasites. And the reason may be that kleptoparasites don't need to locate the spiders to build nests themselves. They actually just need to find another pompilid that has already found a wasp. So the sorts of signals and cues that they need to be sensitive to are likely to be far less, and then selection would favor those species that have relatively less investment in those traits that are very energetically expensive. Moving away from parasitism, now looking at the spider guild. First, let's look at diurnal versus nocturnal spider hosts. We found that pompilids that parasitize only nocturnal spiders had significantly smaller compound eyes, but they had higher density of Sensilla chaetica. And this makes a lot of sense because nocturnal spiders are usually not active during the day, obviously, and we don't know of any pompilids that are nocturnal. So any pompilids looking for nocturnal spiders would have to be looking for them in their burrows or where they're hidden. And they wouldn't be using visual cues, rather they would be using other types of cues to locate these spiders. It is also well known that nocturnal spiders are less visually conspicuous. They have less conspicuous body coloration, and they probably have to find them using non-visual information. Now, if we look at web building, pompilids that use spiders that spin webs have less density of Sensilla ampullaceae and chaetica, and they have more density of Sensilla flacodia. So, oh, sorry, there's a typo there. I said Sensilla chaetica twice. The last one is Sensilla flacodia. So this pattern is interesting because flacodia are of olfactory receptors, and this may be used to detect silk-based odors such as putrescine. So if a spider is spinning a web, the pompilid would detect these spiders by detecting actually the web. In contrast, non-web building spider hosts may use the Sensilla chaetica to detect vibratory behavior that is actually common in spiders that are not spinning webs. Now, if we look at vertical stratification, pompilids that parasitize spiders in vegetation have larger compound eyes, and there could be a couple explanations. So first, this could be a result of divestment because ground-dwelling spiders are more difficult to be visually detected. So selection may favor divestment in the compound eye. With this explanation, however, we would expect to have more investment in antennal Sensilla, and we did not find this pattern. So an alternative explanation is that investment could be related to web building spiders found in the vegetation, and these spiders are more conspicuous, and this could favor investment in compound eyes. So now that was behavior evolution. Let's move on into the phylogenetics of this group. So like I said, there is a phylogeny for the family, and this is a phylogeny that we reconstructed during my PhD and was published in 2015. And this was done in collaboration with Cecilia Weichert, James Spitz, and Carol von Dolen. And we used 5 genes, Sanger sequencing, and we showed that many of the subfamilies are actually monophyletic. We recovered 5 subfamilies, and we sampled 60% of the existing genera. We did not, however, recover good support for the backbone. So we recovered the family Ctenocerinae, branching off earlier in the history of the family. Then we recovered Notosiphyinae, a sister to Ceropalainae, and then Pompiliinae, sister to Pepsinae. And Pompiliinae and Pepsinae are the most diverse subfamilies. They have thousands of species, whereas the remaining 3 have 100 each. So there's a very big difference in terms of diversity here. But because we had this low support, we wanted to know if we use more data, will we resolve these low supported nodes? And also if we increase the sampling for these, will we recover the same subfamilies, or were we missing something? So now we're working on a larger phylogeny of Pompilides, and what we're doing is we're adding more loci using ultra-conserved elements. And this work is being led by Emily Sadler-Pitts and James Pitts at USU and a number of other collaborators. So we also added more taxa. We now have a total of 400 samples, and 80% of the described genera are sampled. And this phylogeny includes a lot more of the Australian taxa. So as you can see, we don't recover 5, but actually we recover 6 of the classically classified subfamilies. So we recover another subfamily that before was embedded in another one. And I'll show a little bit of detail. So here we have the 6 subfamilies, Seropanili in green, Pompilini in red, Tenosarini in magenta, Notosiphyini in orange, Pepsini in yellow, and Epipompilini in blue. So basically what we find is that Tenosarini is actually polyphyletic. And what happened is that we had and sample a bunch of the Australian taxa, which are actually now in the phylogeny, and they're coming out with thin Pepsini. We now recover a sister relationship between Tenosarini and Pepsini, and we recovered a new subfamily that we hadn't recovered before called Epipompilini. And finally, the subfamily Seropanili is the earliest branching, and this is a completely different pattern as before. And it's also interesting because Seropanili is completely kleptoparasitoid subfamily. So here are the phylogenies side by side. And so on the right, we have the old phylogeny. On the left, we have the new phylogeny, and you can see the differences in the monophyly, basically, of the subfamilies. And so what we can actually see is that none of the relationships that were recovered from the 2016 phylogeny on the left were recovered on the new phylogeny, which is on the right. And so the explanation is that really we just added more taxa, we added more loci, and the bottom line is we couldn't really trust those low supports from before. So now let's move on and talk about their biogeography. What do we know? So in 2005, Engel and Grimaldi described a fossil called Bryopompilis interfector, and they placed it as the oldest Pompilid from the Cretaceous, so 100 million years old. But we looked at it closer, and actually it didn't fit any of the characteristic features of Pompilids. So it also didn't fit any of the diagnosis for other Hymenoptera families. So we placed it in its own family, Bryopompilidae, sorry. And so the conclusion here is that Pompilids are not Cretaceous, they're much, much younger. And we have some information on their fossil record. So later on, we looked at the entire fossil record for the family. And on the left, you can see that we mapped all the fossils onto the phylogeny. And what we did was we assigned them to the nodes that would be useful for calibration points in the future. And basically, the black vertical line that you see shows that the oldest known fossil to that time was 50 million years old, younger than 50 million years old. So a couple of years later, literally 2 years later, we found another fossil, and it was actually an older fossil. Slightly older. And this is actually the oldest Pompilidae fossil that we know for the whole family. And the useful thing about this fossil is that it's been dated to a narrow range, so 54 to 56 million years old. And so that's the minimum age that we know for Pompilidae. We obviously know that it's possible that there are more fossils out there, but we just haven't found them. And this means that Pompilids are relatively young from a biogeographic perspective. Thinking about groups that are widespread, you don't expect them to be that young. So the new fossil can be placed in a node that used to be dated from the Miocene, and with this new date, the age of the whole family could be pushed back a little bit in the past as well. But in terms of the historical biogeography, what we know so far are 3 separate studies that we've done for groups that have diverged at similar times, and they have different behaviors. So the first one is the tribe Aporini on the left, and this is interesting because they have they are the smart ones that go into the spider's burrow and sting the spider, so they don't need to make their own nest. And they are found in the Americas and the Palearctic region and diverged 22 million years ago. We then have Ceropalis with an origin 10 million years ago, and they are all kleptoparasitoids. So they're the lazy ones that we talked about before. And they're found in every continent except Antarctica. They're very young. And finally, we have Erigenia, which is 10 million years old, and they're actually parasocial and build mud nests, and they're found in the neotropical region. So you don't need to pay attention to the patterns here because I have summarized them in a single figure. And so here I color-coded the groups and the arrows, and then when the events took place, dispersal events from different continents. So basically, Ceropalis, what happens, they're found everywhere in the world, and their current distribution is explained by multiple dispersal events, some of which happened independently from the same areas. So for example, we have 5 independent dispersal events from North to South America, and independent meaning that throughout the phylogeny, they occurred in different clades. For the tribe Aporini, we find dispersals from North to South America and from the Nearctic to the Palearctic. And for Erigenia, which is only neotropical, we only find that dispersal from North to South America. So it's interesting to see that these groups are not closely related at all, and they have different behaviors, and they have used similar routes at similar times for dispersal. And so interesting things to see here. There's multiple dispersal events. These dispersal events take place from adjacent areas. So there's no long distance dispersal. And there's some dispersal events like the one for Aporini from North America to South America, which took place before the Panama Seaway completely closed, which makes us think that there may be dispersal through stepping stones or in floating objects. And the Panamanian Seaway had a chain of islands that could have been used as stepping stones at the time. There is a temporal and pathway overlap. And again, Ceropalis is very young, found in every continent. And our question is is it because they're kleptoparasitoids that has made it easier for them to disperse this much? So we also want to know if these patterns hold for other widespread taxa with more common behavior patterns, such as in the subfamily Pepsini. And we also want to study patterns at a finer scale. So what is the biogeography, historical biogeography like within, for example, Australia? And this work is being done by a PhD student, Mahin, who is studying a widespread genus called Cryptochyllus. And her talk is on Wednesday at 1:45 p.m. So Cryptochyllus is a genus that is supposedly found in Australia, but there's another closely related genus called Heterodontonyx that is also found here, and they look very much alike. So Mahin is trying to figure out what is the genus level delimitation for this group, and also what is the biogeographic history for it around the world and within Australia. And she has gathered data for ultra-conserved elements using 2 different methods. So she has used hybrid capture, which has been done with our collaborators at USU. And she's also done whole genome sequencing here at ANIC. So basically the rationale behind doing whole genome sequencing is that Hymenoptera have such small genomes, and we're trying to see if we can recover these UCEs using whole genomes, because sequencing is getting cheaper and cheaper. So she put those 2 data sets together, and she sampled most genera within the clade, including the type species of Cryptochyllus. And she also ran some biogeographic analysis, but I will let you learn more about it on Wednesday from her talk. Okay. So now I'm going to switch gears and talk about a different group that it's one of my favorite. I'm going to talk about the mimic revolution in these really, really cute creatures, Mutility velvet ants, just some examples of how stunning they are. So first, let's talk about mimicry. So Mullerian mimicry, which is mimicry amongst equally defended or unpalatable species, produces warning coloration patterns amongst species that are not closely related and share a distribution. This pattern is a result of a strong signal to stay away, which is strengthened by avoidance by predators. And velvet ants display warning colorations, probably due to the very, very strong, painful sting. They're actually in the US, they're commonly known as cow killers because of how painful the sting is. And they have been placed at the very, very top of the Schmidt pain index. In case you're wondering, Justin Schmidt is an American entomologist that has gotten himself stung by a variety of critters and made this beautiful pain index that we now use to rate stinging bugs. So velvet ants in North America show this remarkably clear pattern of shared coloration amongst unrelated species restricted to the same areas. And you can see how in the phylogeny, these coloration patterns are color-coded the same as they are in the map, and they just come out throughout the phylogeny with no pattern of shared ancestry. So this is a very clear example of Mullerian mimicry, what you would expect to observe in spatial patterns and in color patterns. So what happens in Australian velvet ants? So what we know about velvet ants in Australia is very little. There are 261 species described. They're mostly all in a single genus called Ephutomorpha. And basically anything that doesn't fit any other genera has been placed there. There's no taxonomic revision, and the mimicry complexes have not been studied before. The phylogeny has not been resolved, and there is a hint that we have from a recent morphological phylogeny from Brothers and Lelage in 2017, which included 4 genera in one of the tribes, that's in Mutilini, and 2 genera in the tribe Mutilini. And these tribes are circled in blue in the phylogeny. And then on the right, we have the relationships among the genera that are included and 3 genera that were not included in this analysis, which are the famous Ephutomorpha, Astralutina, Ponderotilla, and probably Ephutomorpha was not included due to its very great morphological diversity. And in the ANIC collection, we have about 12,000 specimens, which have been georeferenced through the Digivolve pipeline and have also been the data, spatial data, and the label data has been transcribed as well. And they have been classified into 500 morphospecies, and we find a great diversity of aposematic coloration. So basically, looking at them, we classify them into 4 putative mimicry rings. Basically, the species that are all black with markings that are yellow or red, species that are all red, species that have black head, red mesosoma, and black or dark metosoma, and species that have a red head, red mesosoma, and a black or dark metosoma. And so the question we want to ask is there mimicry within the Australian utility? And this is an endeavor that we've been working for a couple of years with a large group of people. To satisfy this, to answer this question, we need to satisfy these 3 conditions. So first, there needs to be a shared aposematic warning signal. And for that, we're going to do phenotypic trait extraction. So extract the color traits. They need to be not close relatives. So we're going to use molecular data to reconstruct the phylogeny and map the color onto that phylogeny. And we're going to georeference them and look at the distribution to see if they share the same geographic areas. So Claudia Shift, she was a summer student in 2018. She gathered coloration data from 5,000 females from ANIC and the Australian Museum as well. And she performed an NMDS and a PERMA-NOVA analysis. So basically a non-metric multidimensional scaling. And here we see our data. So basically, you see the 4 mimicry rings, and you see how they cluster together, and there's clear structure to the data, although there is some variation within the rings. And then the permanova was significant for difference in color pattern between these groups. But when we look at their distribution patterns, they all overlap. And this is completely unexpected under a strictly coevolutionary Müllerian mimicry process, where all unpalatable species should converge as co-mimics to the same pattern in a single area. So only one pattern should be sustained in the same area. But if mimicry has evolved by Batesian evolution, however, it is easy to see that mimicry rings centered on different models could remain distinct. Or if the mimicry rings were also segregated by habitat, a diversity of mimicry rings could be established. So this pattern that we observed here is not a pattern that you expect from pure Müllerian mimicry. And it could be that either there's a level of Batesian mimicry where some velvet ants are not really as painful as others and are, or actually not painful at all, and are just having an advantage by looking similar to the ones that actually really hurt. The other option is that the different mimicry rings have different niches. So they're being, the predators are being compartmentalized in these niches within the same, what it looks like the same geographic area. So what we did is we looked at the geographic overlap, and we took the environmental layers, we downloaded them for each ring at each point, and we only used the points that had a high degree of certainty in the location. And basically, at a glance, there is no striking difference in these variables for the different rings. But there are a couple variables here that, for example, you can see the one that says the pH. The distribution of this variable is different for every ring. So we want to explore that further, because the mean of each of these distributions is skewed towards a different side. So there could be something going on there, and we're looking into that. Now, for the phylogenetic patterns, this is an exciting new data set that Mari Gianotta, who is a CSIRO PhD student, she got this data set, and we basically received it last Thursday. So this has never been seen by anyone other than Mari and I. And what we did, because we wanted to get a quick tree for today, was we got from whole genome sequencing data, we obtained the 300 most complete single copy genes using BUSCO, and we constructed a phylogeny using IQ3. And here you can see that for this reduced data set, we have really good support values. So almost everything is 100% bootstrapped. Now to the left, we have the genera that are found in Australia, and all the ones that have a little phylogeny to the left are the ones that were included in the morphological analysis. And I have also color coded them with horrible colors, which I apologize. I know that this is not very friendly, but the bottom line here, and the important thing to just conclude from this slide, is that none of the genera, only one of the genera is monophyletic. So Aglaophylla is the only genus that so far we're finding is completely monophyletic. And this is the one in magenta, and I surrounded it with a white square. In the phylogeny, things that are black are males that we haven't assigned to a genus yet. And then color things are females. And then you can see that the famous Ephytomorpha that most of the species are in is completely polyphyletic. It basically shows up in almost every single clade within this phylogeny. So Maddie is going to look at this for her PhD project. And in terms of the color patterns, so we have the 4 mimicry rings at the left, and then the colors that they correspond to. And then we place dots on the phylogeny for this color pattern. So basically, at a glance, what we are seeing is that there's no structure of for these patterns. So they're also interspersed, and this is evidence that there's convergent evolution for these color patterns. And also Maddie will look at this closer for her PhD project. So this data set is very, very exciting for us. It's a large data set, a lot of specimens that we have imaged and we have georeferenced. And this made us think that it would be perfect to use in something like AI ML analysis. So the benefit for generating an AI ML pipeline for the extraction of traits would be very widespread. So what we want to do is be able to extract color, area, size, landmarks, shape from specimens. And these could have applications for new species descriptions, species delimitation, ecological patterns, functional traits, trait response to climate change, and species identification tools. But if we want to extract these traits from museum specimen images, there's a lot of challenges. The specimen preservation can change the color that you observed. The labels are in the way. There's sometimes glue next to a specimen. There are imaging limitations. So there's different lighting, positioning, and background. And how do we make this so that the application of having these traits is broad and useful for the whole community? So what we want to do is, instead of doing what Claudia had to painfully do through some months, look at the specimens, gather information, make a matrix, all manually, and she wasn't able to measure them. We wanted to know if body size was correlated with mimetic fidelity, but there was just no time for that. So instead of that, can we automatically score characters for around 4,000 females that we have imaged from our collections? So the idea is to isolate the head, the thorax or the mesosoma, and the abdomen or metasoma, and gather these color patterns automatically. And then create a matrix that could be used for different purposes. In this case, we want to look at the color patterns. So basically, this is a project that has been in collaboration with Data61. And what we have done so far is being able to isolate the head, thorax, and abdomen using conventional image processing. So this is not AI ML, it's just image processing and manual data curation. Sometimes the labels created were not great, so we had to clean the data. And then with the labels that were very accurate, we used those to train a model so that it could automatically detect the head, the thorax, and the abdomen from our images. And then we extracted the color traits from each body part. Basically, extracted the color of the pixels and reported the main and the secondary colors of each part. And this is an exciting also part of it, because we were able to measure the width and the length of the head, the mesosoma and the metasoma. And I think this could have really interesting applications in the future for use in functional traits. So far, we've been able to score all of the characters for 1,800 females from 340 out of the 500 morphospecies. So this is really exciting. We also got this data a couple of weeks ago. And to determine how different they are from each other, so we got an RGB value for each of the species, the specimens, and we used what is called the Earth Movers distance. This distance is used for color because color distance and perceptual distance are not the same. I mean, perceptual distance is not linear, so you don't calculate just a simple distance between the values of RGB among themselves, like you would do with any other continuous character. So what you can see here is a clustering and then a distance from blue, which is not too distant, to magenta, which is very distant. What you can see is that we don't have a lot of distance between most of the specimens. So we think that we may not be doing this right. We're not expecting this, right? Because with the manually obtained characters, we did get some structure in our data. And then when we did the clustering, again, we got a cloud of points with no structure whatsoever. So now we're looking into new ways of analyzing this data. Do we have to bin different RGB values into colors so that we are not losing the amount of information that we have in terms of how different one color is from another? So this is the big puzzle that we've been working on for some years. And I would like to thank the ANIC Molecular Lab team, which is wonderful. They have helped us so much. And a big shout out to James Nichols too. He has answered all of our questions about sequencing. And a special thanks to the ANIC Hymenoptera Lab. I am so proud, honored, and grateful to work with you every single day. You are amazing. Thank you.
Thank you so much, Juanita, for a fantastic talk. I think we have a bit of time for questions if anybody would like to either post in the chat or unmute themselves and speak aloud.
Hi there, Juanita. I have a quick question.
Sure.
So I was really interested in your pompilid phylogeny that you put together, and you were saying that the Celophalini, the kleptoparasitic genus, was coming out really basally in the tree as kind of one of those first sister clades. What do you kind of think's happening in regards to host use there, coming from a kleptoparasitic kind of genus to non-kleptoparasitic?
That's a really good question. So we don't really know what the ancestral condition would be, right? Because the sister group to pompilids are mutilids, and they use bees as hosts. And they're also kleptoparasites though. So that's interesting there. So it could be that the ancestral condition was just being kleptoparasitoid. I mean, mutilids are a different kind of kleptoparasitoid because they actually go into a bee nest and just lay an egg. They don't really use this thing to paralyze anything, but Ceropalini don't either. And they just lay an egg on a larva, and then the larva feeds on the larva of the bee, right? So yeah, I had never thought about it, but it could be that the ancestral condition was just being kleptoparasitoid, and then some kleptoparasitoid, yeah, the egg and the chicken, right?
Yeah, maybe it's just specialized on.
About it.
I was just going to say maybe just specialized on spider hosts, and from there a whole new world of enemy-free space opened up.
Yeah, exactly. But we don't have that intermediate step of a kleptoparasitoid of some other family parasitizing a spider, right? Like, yeah. Anyway, but that was a good question. So there's some questions in the chat, but let's see. I saw that there was an interesting one. So is there a correlation between venom chemistry and pain? Can one predict even broadly how painful a venom is based on what it's made? And this is, I love this question because I think this is something that we should look into to figure out if there's actually some Batesian mimicry going on rather than pure Mullerian mimicry. So I think that, yes, it's possible to do it. There are some bioassays that are being done by our collaborators at University of Queensland where you can kind of detect how much pain is caused by certain venom. So that could be done. And I think that it's something that would be really useful to be able to tell apart those two processes. I'm not sure if I answered your question, Mark.
I think we've got time for one more question, and then we'll move on to the rapid fire talk session.
Perfect. Can I pick the best one? Well, there's one by James Story. He's saying, I wonder if there's any chance that there's a temporal element to the color mimicry. That's a really, really good question. I have not explored that in depth. I think that we may need to look more like deeper into it. I have done like a quick, okay, where what months do the different color patterns come out, and there's not a clear pattern. But I think that another thing that we were trying to do, and we have a small data set for this, is collecting the mutilids and recording the time of day. That's another one that we want to see, like just some mimicry rings come in the morning and some at late at night. We're also recording what they're nesting on, things like that. So far, we haven't found a clear clue to what's happening.
Okay, great. Thank you very much, Juanita. That talk was fantastic. If everybody could just join me in thanking Juanita again.